well, okay, my management structure, I want like this many postdocs and this many PhD students and subsequently this many master's students and this many undergrads. And so we quickly did some calculations in his head. He was like, so you want like north of 100 people in your lab? I was like, that sounds a lot. I hadn't thought that far. I had just thought about the top-down management structure, how many products I need to work on. And I was like, okay, I really want a student who is able to think that fast on the fly, right? Somebody who's able to kind of also question me in my lovely leadership roles of like, okay, how am I going to, I don't know, establish a lab, right? And Josh has been very collaborative, he's contributed a lot to the way in which the lab runs. I will say that the lab would not run the same way if it was not for Josh. I will say that the work that we've done in the lab has been really influenced by Josh. Josh is a mentor to all of the subsequent students that I've hired and the undergrads that he's worked with. There are undergrads that are now working out in the field that also would like to acknowledge Josh. So like that is a testament to Josh's dedication, his quick ingenuity, and also his skills as a mentor. And so with that, Josh, I will let you take it away.
Thank you so much for the kind words, Dr. Jones. Okay, thanks everybody for coming to my dissertation defense today. I was going to make a joke about how, oh, it's first day of classes here. If you're in the wrong room, you should be belonging somewhere else. But I recognize everybody in this room, so I know you all belong here. So I'm just going to ask that people hold questions until the end so we can keep on schedule. We have a pretty tight schedule, but there'll be plenty of time at the end for questions. So honestly, the pictures in this title slide are nice, but okay. I'm sorry, I just realized that every time somebody joins the meeting room, I lose my script. So I guess I'm just gonna wing this. Okay, so honestly, the pictures on this title slide are nice, but that title does nothing to tell you about what I did the past 5 years and give you a sense of what I'm doing. And so to understand what I am doing, we're going to start with the broader area of research that my research falls into, which is bacterial biofilms. And so to understand what a biofilm is, it can help to start with what it isn't, a planktonic bacteria. A planktonic bacteria are bacteria which exist floating around in, say, water, living as individual units of life, like you or I walking through a busy block in Manhattan. And yes, plankton from SpongeBob is a good example of a planktonic microbe. This can be compared to a spatially organized community of bacteria, which we refer to as a biofilm. Now, these biofilms display some emergent phenomena that isolated or planktonic bacteria don't. There are a large number of these properties beyond what we have time to talk about here. For the purposes of our talk, though, we will mainly be focusing on these two properties, the extracellular matrix and spatial heterogeneity. The matrix is like a net that forms around the biofilm. And it acts a bit like Jello, holding the whole thing together as water fills in all of those pores. And then spatial heterogeneity is when the interior of the biofilm is not uniform. And that's what we will be using for the term spatial heterogeneity. And so that would be like if there were more bacteria here as opposed to out here. All right, so that's biofilms. Now we need to take a bit of a detour to understand why we care about biofilms, which is cystic fibrosis. Cystic fibrosis is a genetic disease affecting the CFTR gene, which affects the mucous lining of the lungs. This leads to reductions in lung function, which affects around 100,000 people worldwide every year, as you can see with this figure on the left, where the patient population per country. Cystic fibrosis is associated with significantly decreased lifespans, as you can see with the numbers on the right, with a drop of almost 30 years using 2020 numbers. For some context, smoking takes about 10 years off your life on average. So this is a significant reduction in both quality of life and lifespan. Now, it's not all doom and gloom on the cystic fibrosis front. A new medical treatment that's come out in the past few years called Trikafta, which is a cocktail of what are called CFTR modulators, has shown incredible promise in treating people with the underlying causes of cystic fibrosis. But as these people get treatment and live longer, that's changing the nature of the disease. People with cystic fibrosis usually acquire chronic microbial infections in their lungs. Specifically, the older that somebody is, as you can see in this figure on the right, the more likely it is that they're going to acquire a specific kind of infection. Pseudomonas aeruginosa infections. And these Pseudomonas-specific microbial infections are associated with poor health outcomes, such as lower lung function, increased rate of lung decline. So we'd like to treat these infections, right? Well, if they were that easy, we'd already be doing it. So why can't we just treat these infections with antibiotics? Well, we can. However, the difficulty arises when, let's take a Pseudomonas aeruginosa infection specifically, in a patient with cystic fibrosis with a chronic lung infection that presents as a biofilm. And this is why we care about biofilms. In cystic fibrosis patients, these infections usually present as spatially organized communities of bacteria. And that brings us to the other main thing that biofilms have that distinguish them from planktonic bacteria. They are more resistant to antibiotics than a planktonic bacteria. So you might have heard of antibiotic resistance before. We're going to use a slightly different term, antibiotic recalcitrance. And that is just specifically talking about how biofilms are able to resist our antibiotics, whereas resistance is a more general term. Okay, so we have these biofilms in these cystic fibrosis patients that are resistant to our antibiotics. How do we overcome that recalcitrance? First, we need to understand, and that's where scientists like us come in. Now, there are a lot of mechanisms that researchers have found that generally explain how antibiotic recalcitrance forms in biofilms. The one we are going to focus on is called periphery sequestration. The best explanation for this comes from probably the primary paper which provided evidence by Sang and co-authors, published in 2013. So what was the evidence that they presented? On the left, you can see these little bacterial colonies in green. And on the outside of these colonies, you can see these rings of white which have formed, which show the antibiotic that they were trying to allow to penetrate and accumulate inside of these bacterial biofilms. And you can see in the picture below, when you take out the bacteria, you can see it much more clearly. The antibiotic is unable to penetrate the interior of these little biofilms. And so the antibiotic gets sequestered to the periphery of the biofilm, hence the name periphery sequestration. On the right figure, you can see how this periphery sequestration was translated to antibiotic recalcitrance. Here, the green represents the alive bacteria, whereas the red represents the dead bacteria. When exposed to the antibiotic, only the bacteria on the outside of the biofilm were killed, which is where all the antibiotic was able to accumulate, indicating that this is a mechanism by which biofilms can resist antibiotic infections. So, what caused this biofilm to sequester all of the antibiotic on the periphery of it? We don't know. We have some ideas about what's causing it in the scientific literature, like attachment to extracellular DNA, but the matter has definitely not been definitively established. And so, enter me. My goal is to try to understand this periphery sequestration phenomenon. I'm going to approach this problem like a physicist, by understanding how chemical species accumulate in biofilms generally, and see if that helps us better understand periphery sequestration. And hopefully, by better understanding how this problem works, we can also understand how biofilms work in two other important contexts. The first is other bacterial infections that people get that present as a biofilm. And there's a large number of these other diseases, including infections on medical device implants, infections on subvenous catheters, and a number of other diseases where bacterial infections can turn into bioinfections. The other main context that we care about this is in the environment. If you've ever been to, say, a river or a stream, and you look at a rock, and you see there's like this gross kind of film that's formed on the rock, that film is chock full of little microbes that have formed a biofilm on these rocks. And so to understand how a potential pollutant might affect an ecosystem, you need to understand how that pollutant is going to interact with the biofilms that make up a large portion of these ecosystems. And so I said I was going to approach this problem like a physicist. So how do physicists do this? Well, I don't know. I'm an engineer, but I'm pretty sure they define their problem. They start by defining their problem, which we already did. I just did it. All right, what's next? So the next thing they do is they put their scholar's hats on and read all those dusty textbooks and scientific publications to figure out Has someone else tried to solve this problem before? By the way, the answer is always yes. Somebody at university you've never heard of did exactly what you originally proposed in 1993. And frankly, they did it better than you would have. But that's all part of the process. Now you can improve on the methods that they use to do their science. Progress, standing on the shoulders of giants, all of that jazz. So once you've gotten your head around how other people have thought of your problem, the next thing you do is come up with your own idea on how to marginally improve our understanding. That's what I go after in units 2, 3, and 4, coming up with my own approach to describing chemical species accumulation in biofilms and testing whether that idea is any good. After that, I'll walk you through how I took that new tool I developed for understanding chemical species accumulation in biofilms and apply it to our original problem of periphery sequestration of antibiotics. And finally, I will go through how I tried to create an experimental system, which we call our synthetic biofilm, which we hoped could replicate a lot of the important properties of biofilms that we identified in AIM 1 and allow us to test some of our theories in a real experimental system. All right, that's the plan. So AIM 1, let's dust off those textbooks and go through the scientific literature. How have people approached this problem in the past? I'm going to spare everybody an in-depth discussion of everything that we found. There's a written dissertation I wrote. If you really care about it, go there. We will instead go over a lot of the main takeaways. What we found was that it was reasonable to understand how things accumulate in biofilms in the same way that we normally understand how chemical species move in general, what's called diffusion. At a basic level, diffusion is just the tendency of chemicals to fill empty space. And we'll talk more about exactly what that means later. However, to understand how chemicals move through biofilms, we found in the literature that there are 4 important amendments we need to make to this diffusion phenomenon and effects that we need to account for. Now, the ones that I have outlined here, which we're going to go over in the next few slides, are not the only effects that are important that we found in the literature. These are just the 4 that we decided to focus on. So let's go through some of these. The first effect that we found, we refer to as the size effect, which deals with how a chemical moving through a biofilm interacts with the mesh surrounding the bacteria that we talked about. By the way, to clarify some nomenclature, I will be using chemical species, particle, diffusing species, somewhat interchangeably here. While these aren't all the same thing, for our purposes, we're going to treat them the same. The first way that the diffusing species, or just species, interacts with that mesh is shown by comparing these 2 particles right here. This is when a larger mesh or a larger particle can't move as quickly through the mesh as a smaller particle. It's a little like a kid with a backpack moving through a crowded area. He's going to move faster through the airport than his dad, who's lugging around his 2 kids carry-ons. He just can't move as fast through all the crowded obstacles that he encounters. The other way that diffusing species interact with the mesh is what we're going to call the size exclusion effect, which is comparing these 2 particles. This is where larger particles can't get through the mesh at all, which is what you see when we're comparing these 2 particles. That's a little like trying to take the airplane through airport security. It's worth keeping in mind that it's not just the particle size that matters. It's the density of the mesh that matters too, which is shown here on the right. A denser mesh means that a smaller particle is going to be needed to pass through it. Good examples of this from the literature are listed at the bottom of the slide, and all subsequent slides in this section, you can find examples of it at the bottom. Okay, the next effect we're going to go to is a little bit simpler. If you have parts of a biofilm, which your diffusing species can't enter for some reason, they won't enter it. These create these excluded volumes in the biofilm, shown here with the red circles, where on the top of the diagram, they prevent the particles from being able to enter these volumes, whereas in the bottom diagram, you can see that the particles are freely able to enter these volumes. And that's all the volume exclusion effect is. The final one that we want to talk about is the attachment effect. This is where there are certain sites in the biofilm matrix, which the diffusing species can get attached to, immobilizing into the matrix. You can see that here in the diagram with the diffusing species getting stuck to these little pockets that we have here in the biofilm. And finally, while it's not necessarily an effect, we definitely learned from the literature that there's a strong importance of that spatial heterogeneity. When the biofilm is different in different places, that affects how quickly things can move inside of it. As I tried to show in this diagram, if things like the density of the mesh or the density of attachment sites, if they aren't even in the biofilm, we need to be mindful of that if we're going to model how these systems work. And so, wrapping up this aim, we've identified 3 ways that biofilms affect the movement of chemical species within it beyond normal diffusion. The size effect, referred to in other places as the mesh effect, the volume exclusion effect, and the attachment effect, where we also identified that the spatial heterogeneity of all of these are important. We will be using this table moving forward, by the way, for all of our main conclusion sections. Our literature review, which led us to these effects, was not exhaustive, however. Specifically, more work synthesizing the literature on the movement of chemicals in hydrogels, which we're going to get to a little later, but these are just non-living versions of these meshes, could have been done. And we also could have looked at a few more chemicals to get a broader understanding of how chemicals move through biofilms. Also, we could have incorporated more effects found in the literature into our analysis, including things like parameters of water filling the biofilm matrix and hydrophobicity of the diffusing species in the matrix. Okay, so now we know what we want to include in our model of chemical species accumulation in biofilms. Now, let's actually build that model. To explain the model, let's start with where we're going to finish. Our model will be composed of these different parts all put together into this one machine. This machine will take in some inputs, spit out some outputs. And to understand how this machine is going to work, we're going to need to start some basics. Now, there will be 2 times in this presentation where I'm going to be showing math and equations. This is one of those times, it's a bit of a doozy. It will last about 5 slides. I'm sorry if this isn't your thing. I've been told I do a pretty good job of explaining math and physics. So do me a favor, and bear with me. You might be surprised by the end how much you absorbed. Before we get to the variables on the right, let's look at the diagram of the system on the left. We have a hemisphere representing our biofilm. So biofilm equals hemisphere, resting on a solid surface, represented in green here, surrounded by some liquid water. Now that liquid water has some particles or chemical species that we care about, like an antibiotic, which is going to accumulate in the hemisphere over time. Now, the first variable we are going to define is that n with a little subscript t, or n little t. N tells you that this variable represents a number of particles, and t tells you that this is the total number of particles in the hemisphere. So n subscript t is the total number of particles in the hemisphere. Next, we will define the volume of that hemisphere, or the amount of space it takes, using big V subscript t. Next, we will define our first concentration, which we will represent with a C. A concentration is just the number of something in a given unit volume. So here, we are always talking about the number of particles. So the subscript here, T, will represent the total volume of the, the T here will represent the total volume that those particles are in. Next up, we have the VO, which is representing the volume of those red objects. Remember those red objects that are occupying space that our diffusing species can't enter? The volume of the objects is represented by VO. And then, well, you can also define this complementary volume, VI, which is all of the volume in the hemisphere that isn't taken up by those objects. Well, you can see that represented with VT minus VO. And then next up, we will define this Ni, which is the number of particles in that interstitial volume. So that interstitial volume is like all this blue and white space. With those number of particles defined, we can now define a new concentration, the concentration of particles in this interstitial volume. Finally, we're going to define what physicists usually call the porosity, which we're representing this Greek letter phi, which is it's essentially the percentage of your total hemisphere volume occupied by that interstitial volume. And then finally, and this is crucial, we can relate the concentration of the interstitial volume to the total concentration in the hemisphere using this relation. CT is equal to CI times V, or the concentration total in the hemisphere is equal to the product of the interstitial concentration and the porosity. All right, that was the basics. Now let's get to the fun parts. So, you know how I showed that the mathematical machine we are going to build has a bunch of different parts? So we're going to build each of these different parts. I'm going to go through each of them and show you how we incorporate them into the final mathematical equation we use to describe diffusion in a hemisphere. You can see an example of diffusion in this cute little animation, where just by the little particles of this species bouncing off of each other, when given the opportunity to fill any space, they will fill any space available to them. And so for our system, which we assume has no diffusing species inside of it initially, the particle will move into it and start to fill the space. Now, scientists long ago figured out how to describe this process with math, which we call Fick's law. This is the equation that tells us how concentration of particles in the hemisphere changes over time. And by solving this equation, we can figure out these outputs. We can figure out exactly what the concentration of particles is anywhere at any time in that hemisphere, just by solving this equation. Now, how do you solve this equation? Get to that later. But that's the basis of what this model is built on: diffusion into a hemisphere. All right, so next up, we are going to add in what's called the volume exclusion effect, the next part of that little diagram, where we talked, and the volume exclusion effect is going to be incorporated through that quantity porosity, which should make some sense, as the volume exclusion effect was about how impermeable objects prevent diffusing species from entering their volumes. And the porosity is how we represent the amount of that impermeable volume in the hemisphere. And so the specific effect of porosity we will be accounting for can be thought of as the need for diffusing species to avoid impermeable objects, as is shown in the diagram. Kind of like hurdles in the Olympics, they slow down the accumulating particles, which we represent in the math equation by modifying the diffusivity, which represents the speed of the particles. If you think back to that diagram of those particles bouncing around, the diffusivity was how fast the particles were bouncing. And so by having to avoid these objects, go over these hurdles, the effective speed of these particles decreases. And so we modify that with this porosity. By the way, we are going to incorporate this porosity a few times into our equation. And so we're going to differentiate how we incorporate it using these little subscripts d. So in this case, this is how we incorporated porosity to affect the diffusivity of our system. By the way, I also have sources for all of these down at the bottom of where we got the ideas for these modeling terms. The next effect we are going to incorporate through the volume exclusion effect is the effect of porosity on area. And that's the little triangle in that dieback model. So as can be seen in this diagram, the impermeable volumes make it so that there is less room for the particles to diffuse through the hemisphere, or less area available to diffuse through. And I tried to show this in this diagram with these dashed lines, representing that the particle has to diffuse through a more limited area when there are these impermeable objects as it diffuses in. And for reasons we don't really need to go into, that's how you capture this effect in the model. I can't really give you an intuitive answer why that's how you incorporate the porosity to account for this effect, but it's supported by models such as Bear et al. Is in his textbook on the subject of transport of objects in porous media. Finally, the way we are going to incorporate porosity the last way we're going to incorporate the volume exclusion effect is by the effect of porosity on the interstitial concentration we brought up before, which I'm calling the effect of porosity on concentration. This is fundamentally accounted for by these little particles just kind of bouncing off of the solid objects and not being able to enter that space, which makes it so that we need to add in this term, where the concentration on an interstitial volume basis multiplied by the porosity gives you the concentration on a total basis. We're also then going to represent, when we solve our model, we'll be solving it for the concentration on that interstitial basis. The last effect that we will account for is the attachment effect. The way that we incorporate this effect is by adding the attachment sites to the hemisphere, where the particle can be immobilized by these little attachment sites, seen by the particle hitting the attachment site and getting immobilized. And we will use the concentration, we will use the term C subscript A to represent the concentration of these attached particles. This will be incorporated into the model by adding this term, which represents the rate at which these particles get attached to the attachment sites. Our specific form that we're going to use is going to be similar to that used by Van Rossum et al. In a similar situation. And by the way, if I write variables afterwards that have a little parentheses after a variable name, that means that I'm saying, for this example, that the rate at which the particles get attached to those sites is a function of the amount of attachment sites. So that's all that terminology means. Okay, last slide with equations. And then we are going to be done. While this is not a distinct effect, we will be accounting for the spatial heterogeneity of the biofilm in our model like we talked about. We will do this by making the porosity and the attachment site density functions a function of position in the biofilm. What that means is summarized in these diagrams. Or let's take this line. This line indicates that there are less attachment sites near the center of the biofilm than there are at the edge or the liquid interface of the biofilm. And then we can control that distribution by changing this parameter in our model chi here, where if chi is 1, there's more attachment sites near the center, or there's less attachment sites at the center than the liquid interface, whereas if it's negative, there are more near the center, less at the liquid interface, and vice versa for the concentration of bacteria and subsequently the porosity in the biofilm. And you can control that using a different parameter, beta. And so by controlling these parameters, we can control how non-uniform these biofilms are. Okay, that's the math for now. So that's everything that is in this model that we're doing. I left some stuff out that we use for actually solving the equations, which is non-dimensionalizing, using parameters, specifying boundary and initial conditions. That's not really the core of what the model is. The model is diffusion plus 3 effects of porosity and then 1 effect of attachment. Moving forward, we're going to be working with not just this full model I outlined, which I might refer to as the full model, but also some simplifications of that model. These are where we essentially remove parts of the machine to see how it affects the results we get. We will represent those in the diagrams, where if an effect is blurred out, we have removed that effect from the model. And then if it's just the parameter itself, so porosity as a function of area, that means that it's constant or uniform in the biofilm. And then finally, if we have these little if we have these little R's afterwards, that means that we're accounting for how, say, the porosity, the attachment site density, is heterogeneous or is non-uniform in the biofilm. Okay, so that's how we're going to represent simplifications of our model. And so, bringing this all together, you could think of this DIVAC model as a machine. By the way, DIVAC is just an acronym for what we're calling the model, which is diffusion in variable attachment in cross-sectional area. It's just the name we use for the model. And so we can treat these parameters, beta and chi we talked about, which control the heterogeneity, the non-uniformity of the model. And by tuning these like knobs on a machine, we can change the final concentration profiles that we get out, which are how we represent what the concentration of particles is at any given time, anywhere in the biofilm. You can read these plots by looking for the specific time that you care about, and each time is represented by a line. And then depending on if it's closer to this edge, that tells you the concentration near the center of the biofilm. Whereas if it's over here, that tells you the concentration at the edge of the biofilm. Okay? And so you're going to see a few of those graphs, so I wanted you guys to understand what they are. And so wrapping up this aim, from aim 2, we developed a mathematical model or machine for predicting the distribution of diffusing species in a biofilm over time. The model we developed accounts for 2 of the effects that we identified, the volume exclusion effect and the attachment effect, through the porosity and the attachment site density in the model. Some limitations from this work is not accounting for the size effect. This would be relatively easy to do. We just didn't focus on it in this research because a lot of other people have spent time understanding this size. We also could have used a better method of accounting for the effect of porosity on diffusivity using something like, say, the Maxwell relation or the Hinson-Coker method instead. And while I didn't talk about it, we specifically developed this model in 1 dimension by simplifying from the full three-dimensional case. This is a strength in that one-dimensional models are easier to solve, but we necessarily lost some information going from the full three-dimensional description of our system to one-dimensional description. It's also worth noting that this volume exclusion effect that we're accounting for is only important when the diffusing species can't penetrate the bacteria, which limits the overall usability. Okay, so we developed this model. The next thing we're going to do is check, is it any good? Is it useful for describing how things work in the world? So the first way we're going to test this is through comparing our model to results from simulations. So rather than go into the gory details, I'm just going to tell you that we can run simulations of our model system. Specifically, we can simulate diffusion in a system with impermeable objects. The simulation I'm showing you is also in a two-dimensional version of our system. The simulation shown here is blue when there's a low amount of antibiotic in the biofilm at that point, and then it's yellow when there's a high amount of antibiotic or particles at that point. It's worth noting here that we did all this in two-dimensional simulations. We weren't, and we did that because of fundamental computational limits, and we also weren't able to account for that attachment term that we talked about in these simulations. So we'll only be testing a version of this model without that attachment part of the machine that we talked about. So we can now take that model we created for how chemicals move through a porous hemisphere and compare the results for that model to the results from a simulation of that system. So we have, now in this case, we tested heterogeneous biofilm, and we actually had no impermeable volume inside. And so in theory, the only part of the model that we need to account for is that original fixed law I talked about. And when comparing the simulations to what the model would predict, we got perfectly. And this shows us two things. One, our model and our simulator are working as we expect. We get the exact results we expect. And so we can now compare our models, our different versions of our model to these simulations to see what versions of the model do we need to account to predict the simulations we see. So the first thing we want to test is, how does our model work in a system where there are actual bacteria in, but they're evenly distributed, which corresponds to that beta value of 0, a uniform biofilm. Now, we would just test the full model and see how it compares to our simulation. But we developed a pretty complicated model with a lot of parts in it. And what we don't want to do is develop a needlessly complicated. We want it to be exactly how complicated it needs to be to predict our simulations. This is taking the idea behind Occam's razor. The simplest explanation is usually the right explanation. So what is the simplest version of our model that explains our data? Well, not too surprisingly in this case, it's the version of the model, what I've labeled model 3 here, which accounts for both the porosity effect on diffusivity and the porosity effect on area, and does not need those spatially heterogeneous terms with the little parenthesis R there. You don't need to create a model with a heterogeneous biofilm if you're only trying to simulate a homogeneous system or a uniform system. And you can see that on the right here. But you do need to incorporate both these effects, because when you only incorporate one or neither of these effects, models M1 and M2, you don't align your model with the simulation results, which are the little circles. And so we need the full M3 to best predict the simulation results we saw. It's also worth noting here that the phi c is the only term that we need to represent equilibrium. We're going to touch on that again a little bit. Okay, now let's test this in a system which you can kind of see here, where the density of bacteria is actually higher near the center of the simulated system and less dense at the edges of the system, which in our case corresponds with a beta slightly greater than 0. And we're going to test 4 different versions of our model against this simulation. The first is model 4, where the porosity effect of concentration changes in the biofilm, but the effect of porosity on the diffusivity constant, PD, is constant. And we don't account for the effect of porosity on area for reasons we don't really need to get into. For model 5, where the effect of porosity is where we account for the effect of porosity on concentration and diffusivity, they're both heterogeneous. And so then model 6 is where the effect of porosity on concentration and the effective porosity on area are both heterogeneous, and the effective porosity on the diffusivity constant is uniform, and then finally, the full model. And just cutting to the chase, what we find is that we need the full model, M7, to best replicate the simulation results we saw. And it might be a little hard to tell from the graph. You can use these little RSS numbers, residual sum of squares, and the lower that number is, the better this model fit the data. And so the full model had the lowest RSS of 24. Okay, so that tells us that in this heterogeneous system, we needed our full model to explain the simulations we ran. What about for a slightly different heterogeneous system, where now we have more bacteria at the edges, less in the middle, which in this case corresponds to a beta of a slightly negative beta. This, we tested all of the same model simplifications that we did before. And what we actually get out of this is that while accounting for the heterogeneous effects of PA and PD, or the effective porosity on area and diffusivity individually through these two models improves performance, for some reason doing them together actually made our model less able to predict the simulation results we saw. And we don't really know why that happened, but it definitely happened. Oh, and there's the system of deep learning. And so some conclusions that we got out of these simulations is that, and by the way, we're continuing to update our little table here, is that we have a new column, which tells us, have we found any evidence for the need to account for the spatial dependence of the term we care about in our model? And we will investigate a few different lines of evidence over the course of this presentation. At equilibrium, which I didn't show here, what we found was that you do need to account for BC, definitely. And then, but that's the only thing that you need to account for in this system. The phi D and the phi A, which are the effects of porosity on diffusivity and area, are both dynamic phenomena. And so you only need to account for them in the dynamic case. And then we also just did not test the size effect or the attachment effect in these situations. And then in the dynamic systems, which are the results that you saw, we again found evidence of the need to account for this BC. And we got mixed evidence, as you saw from that last slide, about the importance of accounting for both the effective porosity on diffusivity and the effective porosity on. Some limitations of this work was that we were not able to test the size effects, and that we were also not able to test the system in a full 3D dynamic situation, and we were not able to test for the importance of attachment. But we at least have some preliminary evidence of the importance of accounting for at least some of these effects. Okay, that's my second aim. Now onto our third aim. So we developed this model that has some preliminary evidence of being important for accounting for how diffusing species move through biofilms. Now let's actually take that model and apply it to the original system we cared about, which was, can we predict antibiotic periphery sequestration in a biofilm? So the first thing we tested was we took that same model, the DIVAC model, and we added in that attachment term. And now we're going to compare these results in a full 3D simulation. Now, this 3D simulation was not dynamic like the ones you saw before. We're only testing it at equilibrium, again, due to computational resources. And what we did was, rather than showing you all of these different beta values I tested individually, we tested, I'm just going to show you all in the same plot. Similarly, for that attachment site density, or for accounting for the effective attachment, if you remember that chi term told you the distribution of attachment sites in the biofilm, we tested a few different densities of attachment sites throughout the biofilm. And long story short, what we found is that over all of the different parameters that we tested, at equilibrium, our model exactly replicates the simulation. And so that is a good indication that our model is able to extend to 3 dimensions, at least at equilibrium. Okay, so we are now going to take a bit of a detour to a different model that I developed. This is what we're going to call the heterogeneous biofilm model. It really is just a slightly different version of that original model, the DIVAC model. Yeah, I showed some of the differences by having the shapes different shapes. And the only difference is that rather than how we describe the distribution of the bacteria and the attachment sites in these biofilms being straight lines, now they're curves. That's the only real difference. I have the functional forms that we use for these different models up here, but the reason that we moved to curves for this is that you can represent a larger amount of different distributions of bacteria and attachment sites by using curves instead of straight lines. But very important to note, we kept those same parameters. They mean the same thing. All they tell you is how uniform or not uniform is the attachment site density for chi, or the porosity density for beta. And so what do we do when we run this model? What we find, which I'm going to conceptually summarize over here, is that when you account for attachment site heterogeneity, where you can see in purple, these are attachment sites, and blue is not an attachment site. When you have more attachment sites at the periphery, you get more antibiotic that accumulates at the periphery. Similarly, when you have more bacteria, which these antibiotics are not able to get into, there's less space for them in the biofilm. And so when you have less bacteria at the edges, you get a higher concentration of your diffusing species. And so when you pull both of these effects together, you get periphery sequestration. And that's shown in the diagram on the right, where when we modify chi, which is the density of attachment sites, or the heterogeneity of that density, when it's largely positive, and in this case when beta is negative for reasons we don't need to go into, we get periphery sequestration, or more antibiotic forming at the edges of this biofilm. And by the way, we wanted to compare our 3D simulation results and our dieback model to the heterogeneous biofilm model that we did. And we find the same trend, which is that when the chi increases, and in this case, the beta increases, we get periphery sequestration. And these are the same things we saw in that diagram, which is as beta increases, we get more periphery sequestration, or more antibiotic at the edge of the biofilm than in the interior. And so, wrapping up this aim and bringing these results into this table, we did two things. First, we showed that at equilibrium, which is shown here, our model aligns with the 3D simulation results and provides evidence for the need to account for spatial heterogeneity of both the porosity term and its effect on concentration and the attachment term. Because remember, those simulations had the attachment term inside of them. We also find evidence that both the heterogeneous biofilm model and the dieback model can predict periphery sequestration at equilibrium and require both of those terms to do so. And that's shown over here, where we have those two terms, which are able to predict periphery sequestration. We are once again computationally limited by testing our system in 3D, not being able to do it dynamically. And while these results aren't shown here, we have separate results which show that it's hard to go from those beta and those chi parameters and use that to predict our quantitative measure of periphery sequestration. And so that was a limitation of the study. And now going to the other half of this, let's now take that model that we've developed and apply it to some real experimental. And we can hopefully use that to try to understand, can it predict some real periphery sequestration? And to do this, rather than generate our own data, we went to that paper that I showed you before, where that showed how the antibiotic was sequestered to the periphery of the biofilm and wasn't able to kill it. Well, luckily, they, in their published paper, showed these profiles, which we could extract data from, and that showed the distribution or the density of the concentration of the antibiotic in the biofilm over time, which is exactly what our model is trying to predict. So let's see, can our model predict these two profiles? They specifically tested their work against two different antibiotics, one with a strong amount of periphery sequestration, or a lot at the edge, and one with less antibiotic. And so we're going to try to replicate the results, our model, for both of these results. So we evaluated the model gradually by incorporating the different forms of heterogeneity. And we can see this, and we can see, again, we don't want a model that's more complicated than it needs to be, but is the least complicated model that fits both of the data. And so we're going to have a bar chart at the bottom right, quantifying the model fit, by the way, which tells you how good was the fit of the data. We're going to start with the homogeneous biofilm model, aka we don't account for any spatial dependence. We're then going to move to accounting for the spatial dependence of the porosity. We're then going to move to a model which accounts for the spatial dependence of attachment. And finally, the full model, and see which one fits the data the best. So the homogeneous biofilm model, not very good. It was not able to really fit the data well at all. And you can see that in the bottom right, with these bars being pretty high. The lower that number, better. The heterogeneous, accounting only for heterogeneous porosity, also did not do it very well. Once we account for just heterogeneous attachment sites, it's starting to look better. We got good fit for the accumulation of the first antibiotic, but not very good fit for the second. And I can go into later why we think that was the case. But when we account for both effects, we are able to better model both of the antibiotic accumulation that we saw, where you can see this with these bars being the lowest over there. And so what we got out of this aim was that when we compare some of our model to the experimental literature, this provides evidence of the importance for accounting for the heterogeneity of these biofilms when trying to model antibiotic accumulation inside of them. Some of the limits, which I kind of briefly mentioned here, I wouldn't necessarily call it a limit, but the reason that we only got mixed evidence for this phi A, which is the actual term that we accounted for when we accounted for the porosity heterogeneity, the reason that it was only important for the tobramycin as opposed to ciprofloxacin, at least we theorized that the reason that that was important was because tobramycin has a lot of attachment sites. And so if you have a lot of attachment sites, that's going to be the dominant effect, and you don't need to account for the effect of porosity. It's just not as important. And then finally, and then the other main limitations were that we probably should have used a different coordinate system to model our data. We used a Cartesian coordinate system, but we should have used the spherical coordinate system. And we didn't use an algorithmic fitting procedure. Those models and the fits that you saw to the experimental data, those were not hand fit. There was a procedure, but it wasn't algorithmic. And so that could be an improvement. All right, on to our last aim. We did all of this modeling. Now let's try to make an actual biofilm or a synthetic biofilm that we could test this model. So when we're creating this synthetic biofilm to try to test, we want to replicate those three important effects that we talked about. We want the synthetic analog, that synthetic system that we're using to replicate the size effect, the volume exclusion effect, and the attachment effect. And the way that we're going to do this is we're going to use the baseline platform that we're using to build this system are what are called nanocellulose hydrogels. And I touched on this a little bit before, but a hydrogel is very similar to that matrix that forms around bacteria, in that it's like a mesh, but there's just no bacteria inside. And so it's a non-living mesh. And that's what we call a hydrogel, which also has a bunch of water inside its pores. This work was done in collaboration with my lab mate, Daryl Taylor, who's an expert on nanocellulose hydrogels. And so that was the platform. We tested nanocellulose hydrogels that were supposed to replicate that extracellular matrix that forms around the bacteria in a biome. And my aim was to test the transport properties of these hydrogels. So what we then, to actually make these microcaps, you can see an example, a diagram of our microcaps over here. And what we did was we added into this hydrogel mesh that I talked about, we added in these red microspheres. And what these red microspheres did was they prevented our accumulating species, in this case, these nanodextrans, from entering these regions of the microcap. At least that's what we theorized. And so these microcaps, or these little microspheres, were how we accounted for the volume exclusion effect in our experimental system. To account for the size effect, we use that nanocellulose hydrogel, which acts as a gel and should be able to impart that size effect we talked about. And then finally, to account for the attachment effect, we used microspheres with varying charge on their surfaces. And by varying the charge on their surface, if we also use different charged nanodextrans, in theory, oppositely charged, if the nanodextran is positively charged and the microcap is negatively charged, in theory, they will attach to each other. They'll get stuck and immobilized on each other. And so that's how we try to account for the attachment effect of our system. All right. And so here's an example of what our experimental apparatus looked like if you really zoomed in. what you have here are these microcaps, where we have a water solution that has a high concentration of our accumulating species, in this case, what we call nanodextran, flowing in, and it has these little green labels on it so that we can see them with a microscope. And so then as they accumulate inside of the microsphere, they will hopefully, and they will gradually accumulate inside of these microspheres. And we can take images of this microsphere over time using a microscope. And this microcap is sitting on this coupon. And you can see an actual image that we took here, where we have the blue representing that hydrogel mesh, and then the red being those microspheres that are meant to represent bacteria embedded inside of that hydrogel mesh. And then that green here represents that coupon sitting on top. Okay, what we're then going to do is we're going to use this confocal microscopy, which is essentially a 3D microscope, to measure the nanodextrin concentration accumulation over time. And the way we did this is shown on the left here, where we made our nanocellulose hydrogels, where we added a bunch of fluorescent microspheres inside of it, made a bunch of little microcaps, and then put them inside of this flow cell. And then we hooked up this flow cell to high concentration of nanodextrans that we cared about. And then we would flow those nanodextrans into the system while it was on a microscope. And then we would be able to take images. And you can see an example here. Yeah. And so this is what happens. We first took an image before any nanodextran got inside of it. And then you could see the nanodextran accumulating inside of the microcap. And at maybe like this time point, we would then take what we called an accumulation image. So we had an image of the microcap before anything got in, and then after it got in. And so we could use that to figure out the concentration of the nanodextrin inside of that microcap. By the way, we're also not going to directly use the concentration. That would actually be pretty hard to figure out. We're going to use what's called a partition coefficient, which is just the concentration of the nanodextrin that we see in the microcap divided by the concentration in the surrounding water, because that's a little more directly measurable. Okay, so we have this microcap. Does it replicate the size exclusion effect of the pair? And here's what we're looking to see. We expect larger particles not to accumulate in a mesh of a given size. And so when we use the same hydrogel, but flow in a small nanodextran versus a large, we should see lower concentrations. And that's exactly what we saw. Our microcap was able to replicate the size effect that we cared about. What about the volume exclusion effect? Well, don't look at the title yet, but the nanodextran, and remember, this is the nanodextran not being able to enter these microspheres. And so in theory, if we don't have any microspheres in our hydrogel, we should get higher concentrations of this nanodextran because it's able to occupy all that volume. Well, it didn't work. Our microcaps were not able to replicate the volume exclusion effect we cared about. How can you explain this? There's actually probably a very easy explanation. We probably just didn't use a high enough concentration of these microspheres. If you have more microspheres, you're going to get more volume excluded, and so you would expect the concentration difference to be high. And we probably just didn't use enough to see the effect. And then finally, and I'm not going to walk you through, it's not intuitive how to really interpret some of this, but the important part is that when we tried to test the attachment effect, that didn't work. We didn't see evidence of the attachment effect from the data that we. But this can also be explained fundamentally by not having enough microspheres. If there wasn't enough surface, enough sites for these nanodextrans to glom onto, you're not going to see a strong attachment. And so, bringing together the conclusions for AIM 4, the microcap features that we cared about have an equilibrium. We were only able to test this system at equilibrium. And because we tested at equilibrium, the three effects that we only expect to see dynamically were untested, or we were unable to test. But we saw evidence for the size exclusion component of the size effect, and we did not see evidence for the volume exclusion effect and the attachment effect we expected to see. And we were not able to run the system dynamically. Some other limitations of this is that these are synthetic biofilms. They're not real biofilms. And so hopefully, in future studies, we could use real biofilms to test this system. And then it's also worth noting, and this is a little in the details, but the nanodextrins that we use, I showed them. It's just like little individual guys running around. That's not really what they are in solution. They tend to kind of glom onto each other. And we actually had a bit of an issue. They were glomming onto each other a little bit too much, and they should have been more dispersed in our system. And so, bringing it all together into the final conclusions in one big table. Let's not break down each thing in this table. Let's take a step back. What does this all mean? What we've done is we developed a model which generally agreed with simulations of diffusion in these heterogeneous biofilms that we were testing, and generally explains the periphery sequestration observed in literature experimental data shown through, and it's able to do this by modeling the heterogeneity of our system. And we did develop an experimental system that has currently unrealized potential to synthetically replicate important biofilm transport properties. And while I could, of course, could have investigated more of this table, PhD, like all things in life, must eventually finish. Okay. So I think I will, what time is it?
2 minutes.
Okay. I'm going to skip ahead a little bit because I'm hoping to catch my dad. Ignore these for now. I'm skipping ahead to the family side. All right. I want to, so in the acknowledgement section, I will start with thanking my family members. They have made me the man I am today, and I could not be more thankful for both my parents, for their love and support, and endless, and like really just being the rocks that I can always rely on in life. I want to thank my sister Megan for being a great older sister who I always looked up to, and whose like love and care is something I always hope to replicate in my own life. And also my little sister Jillian, who I also admire for her dedication and determination and really caring about her love relationships. Sorry if that was a little rushed, but I wanted to make sure Dad got the family section. Okay. He has to leave at 11. Okay, we're now going to go back to the front. Okay, this is normally where you start. I wanted to thank a lot of my former I'm going to start by thanking a bunch of my former teachers, specifically Dr. Happel, Dr. Weber, Mr. Raff. You guys were all very important for my development as a scientist upon reflection. I also want to thank a lot of my former mentors like Madi, or Dr. Malmali, Dr. Bindra, Yusef, and Nick. You guys were all super important for me developing as a scientist and showing me good examples of how to be a good scientist and good people. I also want to thank the ITEP program and all of the people in it, from the faculty to some of the students who are here today. You guys were awesome. The ITEP program is how I was able to explore my passion for environmental science, but you guys didn't really get to see much here. And also how I got the training I need to do my job that I'm starting in 2 weeks, being an environmental exposure scientist. I also want to thank the Northeastern Chemie Department, which Dr. Jones mentioned is where I started my PhD, and all of my friends who I had there. You guys were great friends. I also want to thank the Duke Environmental and Policy Clinic, who really helped me explore my care for the environment and helped me learn how to actually be an environmental scientist and what that means. Specifically thanking all the people who helped run it when I was there, Reich, Michelle, Lee, Nancy, and my partner on my project, Nate. I also want to thank people from the CE department who helped make my PhD possible, like Dewana, Rocky, Dr. Shikim, and Ruby Nell. And I want to thank all of my committee members who so graciously gave me so much of their time, like Dr. Gunch, Dr. Wisner, and Dr. Stewart, and helped me guide and make this dissertation what you saw today. And also, I have funding sources like NIH in 2 ways, and then those are the departments that I also was supposed to thank BioVector there. They always say you have to cite them, so there it is. This is all of my friends who I've made at Duke, and a bunch of fun pictures of us together. You guys have been great. You have made my experience at Duke so awesome, and I couldn't appreciate all the friends and support that I've gotten from you guys over the years. I also want to thank all of my friends from New Jersey, Minnesota, and home. They have the most pictures because they've been my friends the longest. And we've just had so many fun times together. You guys were always the rocks that I could rely on when I didn't want to be in academia. And that was very often. You always kept me grounded. I also want to thank all the members of the SEAL lab. I'll go through each of you. I want to thank Andrew, Daryl, Elizabeth, Chelsea, Kayla, and our newest member, Georgie. I also want to thank Sarah Kate, who was so important for my research. early on when I was in. And, but, probably most importantly, I want to thank Dr. Jones, who has been a great leader, or I believe he's been a great leader for our lab for for some very hard times. We've been through everything. We've been through the lab moves, the university moves, the changes in research directions, and he's always been a steady, reliable presence through all of it. And perhaps most importantly, he's been a great person to develop good ideas. And that's mostly what I believe PI is for. I mean, maybe that's just my opinion, but he's helped develop this project and made it a lot better. And I really appreciate him for all of that and everything else he's done, and modeling how to be a good scientist in a Next, I want to thank my new family, who I've gotten in the past few years. Thank you, Susan. Thank you, Tom. Thank you, Chris. You guys have been so welcoming for me as part of my new family. And I call it new family now, but soon it will just be family. And you guys have been so warm and embraced, and it made me feel loved and in a support system here in North Carolina that I could always count on. This is the family stuff I already talked about before. I want to thank my pets. We have 4 of them. We have 3 cats and a dog. And they're all wonderful. And they're always important for me to go home and remember that I am not just a PhD student. I am also, and there was always love and support at home every time we would go there. And thank you, Evie, our dog, our unfortunately a little dumb cat, Nico, our very zesty boy, and our little demon, Chateau. Thank you to all of you guys. And then, last but not least, my fiance, Sam, who has been so instrumental for me during my PhD, and has been just an amazing part of my life, brings out the best in me. I hope I bring out the best in you too, and has just made my time here at Duke, and hopefully for the rest of my life, a life very much worth living. And thank you so much for all the help. Okay, now it's time for questions.
Might be super nitpicky and maybe just me misremembering my organic chemistry. But when you were testing your synthetic biofilms, you were using amino and carboxyl groups as your charged groups.
Yes.
I could be misremembering organic chemistry, but are those groups charged enough to have had the attraction that you were hoping for? Because I remember them being pretty charged.
Yeah. So at the pHs that we're testing, all of the functional groups that were present on the let me get to a good slide that shows this. That would be like, what we're talking about is how much charge is on these surfaces. For the positive and the negative, all of the groups that were present at the pHs that we tested should have been charged. And so in theory, all of the groups that were there should have acted as charged to try to attach the nanodextran, but it's very possible that the concentrations just weren't high enough to really attach to those nanodextrans. It's definitely possible that even if we use a higher concentration of microspheres, we won't see an attachment effect.
So it's something you could alter in the future.
Yeah, there are a few ways we could try to tackle that in the future. You could actually take this hydrogel mesh. This is actually originally why we used nanocellulose. You can directly change the charge of this mesh by doing some chemical transformations to it and adding charge directly to it. And so that's one way you could try to add attachment sites to the system. We did it through the charge of the microcaps for reasons we don't know.
I remember you during your ITEP seminar saying that for microcaps, you would individually put each one into the hydrogel.
Yeah. Still doing that.
Yeah. Now hearing the explanation that you didn't see the effect, you didn't have enough. I'm wondering, A, what was the concentration of microcaps in the hydrogel? And then B, at what concentration do you think you need to get to C?
So I'm going to just aggregate that question a little bit. So the way that I made these little microcaps is I would fill a syringe with the liquid version of them, and I would just kind of poke that coupon that they were sitting on top of a bunch, and then there would be a little residue behind, and then the residue would dry, and that's how we would make these microcaps. And then and so that's how we made them. And so that didn't really affect the actual concentration of microspheres inside of them. Now, what concentration of microcaps might we need to use to get to high enough levels to see the effects that we wanted to see? Right now, we're using a very low percentage, like 0.01%. You could probably need to take that up to probably about 1% before you start really seeing the effects. You can't go that high, though. There is a limit to how high of a porosity you can use in our system. Eventually, you just can't pack the spheres in. And so there's this like middle ground, which I could hypothesize, and I will talk with Georgie about when she picks up this project, about where good middle ground to start testing. Good question. Both good questions, by the way. Let's see. Oh, just congrats.
So the application of this in the bigger picture is to help cystic fibrosis researchers fully understand what antibiotics could be doing in biofilms, or at least that's one of the applications.
Yes.
What would you, if you were in front of a panel of like the top cystic fibrosis researchers in the country, what would you tell them is the biggest takeaway from your work?
The biggest takeaway from my work is cystic fibrosis. Well, one more study is needed, but probably the biggest thing I would tell them is that if you're looking at new therapies and you're going to test them against actual biofilm systems, if you can, try to see what the distribution of your antibiotic actually ends up being in the biofilm. Don't just do a bulk test of efficacy. And if you're going to do a bulk test of antibiotic efficacy, do it on a biofilm. Don't just do it on the microbe itself. There are lots of tests that you can do for that. And we actually, at our own lab, have our own new novel platform for testing not just microbial efficacy, but looking inside of the biofilms to see how these antibiotics are getting inside of it. Thanks to Sarah Kate, who I mentioned before, who developed that system. And Ashan, my undergrad, who's picking up and working on that system.
Is there any attention paid in antibiotics currently to when they're developed and like size and all that kind of stuff, or are they just not?
Well, there's only so many antibiotics that develop. And so you're really asking about a very limited number of cases. And for the most part, when you're developing an antibiotic, and there are good reasons they do this, and they should do this way, they're mainly worried about, can you kill the bacteria at all with it? Then maybe they might be concerned about, could I kill a biofilm version of the bacteria? Luckily, there are a lot of antibiotics on the market. And so what you could do is if you have somebody who has a specific biofilm infection, you could, in theory, test the bacteria, the antibiotic that you're considering using against the biofilm, not just the antibiotic itself. But it is worth noting that people have tried that before, and the efficacy, the usability of that is not always super clear. Like you can test antibiotics. So what will usually happen is like a doctor has a patient and they're trying to determine what antibiotic to give them. And so they'll take a sample of the infection and run a bunch of different antibiotics that they're considering against those bacteria. And when you try to, and then whatever one works best, that's the antibiotic you give the patient. When people have tried to do that test not on the planktonic bacteria, but on the biofilm bacteria, there's mixed results about if testing against the biofilm versus the planktonic actually improves the antibiotic that you use. And so more research needs to be done. Good question.
You mentioned you were having trouble fitting, or you were trying to come up with an algorithm to fit your model. And from what I understand, it's like a physical model, you're trying to fit data, right? What was your approach to fitting that model, and how do you think it can get improved?
So the approach was me sitting down and manually testing a lot of the parameters.
Grad student
grad student descent into madness, sitting there and manually changing the values a bunch and testing, okay, this made the model better or fit better or worse. If I were going, so the fundamental issue you have here is that the equations that I'm using are partial differential equations, and it takes a long time to solve a partial differential equation. And so trying to do it algorithmically is hard. Like just to run the model once is a few seconds. And usually to fit data to a model algorithmically, you have to run it thousands of times, not millions of times. And so doing that obviously would take a long time. Ideally, you could solve the partial differential equation faster. And there are a lot of ways that people have come up with for how to solve those equations faster, like physics-informed neural networks, which I did try my hand at some point. But I never was able to really get it to work. But in theory, you could use those more advanced techniques of solving partial differential equations to solve your model faster and fit the equations algorithm. I see there's a question on Zoom. Talia, do you want to say your question? I think, Talia, is your hand raised or? Oh, never mind.
On that note, if there are no more questions, we'll dismiss the public audience portion of this. We can give Josh one more round of applause.
Thanks to everybody on Zoom.